Hello and welcome to Chapter 14, Part 4, and this part really focuses on mutation. Now, we have already discussed mutation in detail when we were discussing the DNA chapter. And so why are we bringing it up again in this chapter? And the reason is because while mutations occur in the DNA, the result is in the polypeptide. And that's the phenotype, right? So we're going to take a look at mutations again, this time with a little bit more emphasis on what happens at the level of the protein. So just kind of a reminder of a few terms, and a bunch of this information in this part is review. So mutations are changes in the genetic material of the cell. So changes in the DNA of some sort. And we've talked about many different kinds of mutations. You might remember point mutations. And a point mutation is any time where a base is changed from one nucleotide pair in a gene to a different nucleotide pair of a gene. Now, it's really important that I'm emphasizing gene right now because it's genes that make proteins. So a change in that single nucleotide strand can, in fact, lead to the production of an abnormal protein. Now, depending on where this happens, it can have really important differences. For example, if the mutation happens in the germline, and by germline we mean if it happens in the egg or the sperm, if it happens in the germline, then when that egg and sperm meet up and fertilize and form the embryo, that embryo is going to have that mutation in every single cell of its body. And as the embryo develops into a whole organism, it will still be in every cell of that individual's body because the DNA has the mutation, and as cell division, mitosis has happened, you pass that on. On the other hand, if you think about the next generation, in the next generation, only half of the sperm or eggs, only half of the gametes will have that mutation. Why not all of them? Remember that when you make gametes, you separate homologous pairs, and it's very, very unlikely that both members of the homologous pair have the same mutation, especially if that mutation happened in just the sperm or the egg that met to form up this embryo. So only half of those gametes are going to carry the mutation, but I could say it another way and say, well, half of the gametes are going to carry the mutation. So that's a pretty big percentage. It's fully 50%, right? On the other hand, you might have a somatic mutation, and a somatic mutation happens at some point after fertilization, but before you're finished developing. So in this case, what we have is a mutation in the embryo, and it happens kind of in the shoulder region. Not sure why they picked the mutation there, but that's where they picked it to be. So in that somatic line, that only the cells that develop from that original somatic mutation are going to have the mutation. And because they're not in the germ cells, none of the gametes will carry that mutation. This means that you cannot pass that mutation on to the next generation. So the point here is that if a point mutation happens in a gamete, it's got a 50-50 chance of being passed on if that individual makes it to adulthood and reproduction. And if it happens in a somatic part of the body, a non-germline location, then it will only affect a small region and will not be passed on to the offspring. Let's take a look at a very specific point mutation that happens that causes a very deadly disease called sickle cell. And we've talked about this before, but just wanting to show you what's going on here at the level of the DNA, the mRNA, and then the protein. So this is we call it the wild type. Remember that we use that word wild type to refer to any of the most common types. It's not necessarily dominant or recessive, it just means more common. At a particular location along the wild type DNA, we have these 3 letters in the coding and template DNA, and the GAG, so that would make this the template and this the coding strand. GAG in the mRNA, and that codes for glutamic acid in the sequence of the protein, the amino acid sequence of the protein. If we have the mutant type, instead of it being CTC, it's CAC in the template strand, which makes it GUG in the mRNA, and that changes our amino acid to valine, and that turns out to completely change the structure of the hemoglobin molecule, and that leads to the deadly disease of sickle cell. Just a reminder that sickle cell disease changed the shape of the hemoglobin molecule, which causes the change in shape of the red blood cell. And this change in shape makes the red blood cell diameter, like a little bit longer, and also causes it to kind of wedge into the capillary system of the individual who has the disorder. So that's one little point mutation causing a huge problem for the individual who has it. Other kinds of small-scale mutations that we talked about before were the point mutation that I just talked to you about, actually, I just reminding you of, is a nucleotide substitution. But we can also have insertions or deletions. And so this is a kind of nice example of what happens with the substitution versus insertion and deletion. Just kind of remember that this is review for you. So with a substitution, we change one letter. In this case, this C right here gets substituted in for an A, and it changes the bases. But if you count up the bases in this little string of bases right here, sequence, there's 9 of them. If you have an insertion, here we're subbing in the letter A, the nucleotide A, and we add a base pair. And so we have 10 bases. With a deletion, we remove a letter. Here we're removing that C that I mentioned before over here. And now we only have 8 bases long. Again, these are small scale point mutations. The example that your book steps you through is this one right here, and really it's just things that I hope that you're familiar with. A nucleotide base pair substitution, replacing one base pair for another. In this case, what we have is a silent mutation. And a silent mutation means that you don't end up having a change in the codon. So if you look up this codon right here, GGU, and you look up the codon GGC, you get glycine each time. So that's good. You can have a mutation and not have any effect on the phenotype. Another kind of substitution ends up in a missense mutation. That's where you change one amino acid for another. So in this case, remember we had glycine before. In this case, we're substituting this T and instead of a C, and that gives us an A in that first letter of the codon. Remember, sometimes in the third letter of a codon, you can have some wobble, and it's okay, it still codes for the same amino acid. But if you're changing the first one, you're almost always going to get a new amino acid. So when you get a different amino acid, you have a missense mutation. Substitution mutations really are more often than not missense mutations. The next one is very interesting. It's a nonsense mutation. A nonsense mutation happens when you change an amino acid codon into a stop codon. So what we're doing is we're changing our second amino acid in this sequence, and the mRNA was AAG, but we're substituting that first A, and now it's UAG, and UAG is a stop codon. So what that happens with that is that when the amino acids are coming in, the tRNA reads a stop in it, and it brings in that stop tRNA, and that's the end of the making of the protein. So that can be a very damaging thing. It almost always leads to a non-functional protein. It's because the protein is much shorter than it ought to be. Now, insertions and deletions, I just went over with you, addition or loss of a nucleotide pair. And these mutations often have just disastrous effects on the protein. Often just way worse than substitutions. Remember, with a substitution, unless it's a stop codon, a missense, you can have these silent mutations. You can have your missense mutations, but if you have that, then it might not be so bad. Or on the other hand, it could end up in something like sickle cell disease. But with an insertion and a deletion, you are shifting every amino acid after that mutation. You're shifting the reading frame. And so we call those frame shift mutations because you're shifting the reading frame. Before, when I said this, you didn't know what a reading frame was very well, and now you do. So here's an extra A put into this sequence, and just by putting in that extra A, you have made a stop codon. And so, well, that means everything past that doesn't get put in anyway. But if you have a frame shift mutation where you delete a letter, every single amino acid after that place where the deletion occurred is going to be in a different reading frame, and every single one is going to be different. Same thing, it can be worse early on, and if you delete a whole if you could delete 3 of them in a row. And this does happen. For example, one of the common mutations in cystic fibrosis is a deletion of 3 letters in a row. So if that happens, then you don't get a frame shift itself, but an amino acid is missing, and a whole amino acid is missing. And so you do shift not the reading frame, but you do shift the amino acid sequence. So that would not be good. Also, just as a reminder, that spontaneous mutations occur during DNA replication. They can happen during recombination in meiosis. They can also happen during the repair process. This is a review for you. A mutagen is anything physical or chemical that causes a mutation. But spontaneous mutations happen even without the presence of a mutagen. They just happen because the process of DNA replication is not always precise. The process of recombination in meiosis is not always precise. The process of DNA repair is not always precise. So they always happen. But if you're exposed to mutagens, the rate of mutation is increased significantly. We know that ultraviolet radiation is a source of mutation, so doctors recommend that you not get a sunburn, not expose your skin to too much UV radiation. X-rays, of course, can cause mutations as well. We learned that earlier. We saw that really cool example of that earlier in this lecture where we talked about mutating fungi. Chemicals like cigarette smoke, nitrates and nitrite preservatives like those found in hot dogs and red wine, barbecuing. It turns out that if you burn food till it's like got that nice crust, that crust is a mutagen. And benzoyl peroxide, which is common in acne products, is a mutagen. There are some viruses and other infectious agents that are known to cause mutations, including the very famous HPV. The vaccine for HPV was kind of put into play about 15 to 20 years ago, and the rate of HPV transmission has decreased, and along with that, the rate of cervical cancer is starting to decrease. We're seeing a lot less people transmitting HPV, which is an agent for cervical cancer. Also, Helicobacter pylori, which is a bacterium found in the stomach, and it's passed through food. It can lead to stomach cancer. So, kind of just categorizing those. Most cancer-causing chemicals, carcinogens, are mutagenic, and most mutagens are carcinogenic. So when we talk about mutations and mutagens, we often just go right into thinking about cancer because, of course, that does affect our daily lives. Also, and luckily, we do have methods for testing mutagenic activity of chemicals. So we know that for sure that cigarette smoke, nitrates and nitrites, barbecue, these things, they are mutagens and best to be avoided. So I wanted to spend the last bit of this lecture talking to you about the concept of the gene. We've already spent much of this chapter assuming what a gene was, but I want to broaden that out just a little bit for you. I want to kind of make you think about this. The definition of the gene, the word gene, has changed throughout the history of genetics. In this lecture alone, even though I didn't really name it, I have discussed the definition of gene as, first of all, a unit of inheritance. We pass down our genes, right? I've also talked about it as a region of a nucleotide sequence on a chromosome. I've also, especially in this lecture, talked about it as a DNA sequence that codes for a polypeptide chain. But I don't want to leave this lecture with just those three definitions. There is a fourth thing that's also important for you to understand, and that is that a gene can be defined as a region of DNA that is expressed to produce a final functional product. And that final functional product can be a polypeptide for sure. That's what we spent a lot of this lecture talking about. But it can also just be an RNA molecule. And so if it's just an RNA molecule, we talked about ribozymes, right? tRNA is a final product. Ribosomal RNA is a product that joins up with some protein and makes ribosomes, right? So RNA can be the final product, and so therefore we would have to talk about the DNA that codes for that RNA as the end product there as a gene. That DNA that codes for transfer RNA, that's a gene. And you've got 64 different genes because we do have 64 different versions of transfer RNAs. Okay, so that is my lecture for you on chapter 14. I hope you have learned a lot. The main things, the main processes of transcription and translation can be challenging. I hope that you have picked up the basics and are going to just get something out of it. All right, see you later.